All right, in section 22.9, I'm going to talk about mutations to the genetic code. And basically, a mutation is a change in the nucleotide sequence of the DNA. Some mutations are random, while others are caused by a mutagen, which is like a chemical of some sort that affects our DNA. And a point mutation is the substitution of one of the nucleotides for a different one. So for example, if this is the original, our DNA code, and we were affected of some sort with a mutagen or something, it just happens, it's instead of a G in one place, you would get a C. All right. So there are several types of mutations. One is a deletion mutation, when you actually lose one of the nucleotides in your code. So instead of having a GAG and a TTC, you actually lose that G completely, and now the A and the T are hooked all together. Another one is an insertion, which obviously is the opposite of a deletion, is where you get an added um, one instead. So a nucleotide is added. So in between the G and the T, you get a C in place of it. All right. Um, Types of mutation. I'm sorry. And if you do have a mutation, occasionally it might not have any effect whatsoever. All right. So, and that'd be called what's called a silent mutation, where maybe you do have a point mutation where one of them is changed into a different um, nucleotide, but the amino acid that is coded for that codon would be the same. So like in the C and the TT case, on the messenger RNA, it'd be a GAA, right? It's the opposite, GAA, which is code for um, a glutamine, all right? But if you have a CTC, which would give you a GAG in our messenger RNA, that's still a code for the glutamine amino acid. So even though you had a mutation, it actually had no effect on the amino acid. Okay, a mutation that produces a protein with one different amino acid usually has a small effect overall. Okay, so if you have a change, and instead of having a glutamine or a glutamic acid, you have a valine or a valine in place of it, and if it has no really change or alteration in the amino acid, then it's really not that much of a big a deal. All right. Some proteins, however, such as hemoglobin, if you just change one amino acid, can actually have a big issue. All right. So it just all depends on what amino acid it is, and it depends on what protein it is. Okay. If a mutation causes a change, like producing a stop codon, okay, the remainder of the protein will not be synthesized. That can have a huge effect. All right. So just because you have one amino acid change, it might not be a big deal, but other times it could be a big deal. All right, because if you change a C for an A, all right, instead of having CTT, you have ATT, and UAA in the mRNA is code to actually tell the building DNA strand to actually stop building, or a protein to actually stop like building, and it stops it. So that protein will never be made again, and that could cause a lot of issues. All right, so it just all depends on the type. All right, when a mutation causes a protein deficiency or protective protein synthesis, and this mutation is passed through generations and generations, is considered a genetic disease. And here are just a few of them. There is like Tay-Sachs and sickle cell anemia, and a few others. You can kind of see some. All right, 22.10, they talk about recombinant DNA. And this is basically synthetically made DNA, all right? What you do is you use the cells of a person, and All right, so recombinant DNA is a synthetic form, and it uses live cells to actually make the DNA. All right, so there are three key elements that are needed to synthesize DNA. One obviously is a DNA molecule, all right, in which a new DNA segment will be inserted. Two, the enzymes, okay, specifically an enzyme that cleaves the DNA at specific locations. And finally, a gene from a second organism that will be inserted into the original DNA. It's kind of like you hijack the genes of another organism. Okay, so here's the whole picture. Okay, but they kind of slowly go through this. All right, first, what you do is you take the DNA from a bacterial cell. Okay, and it's cut by what's called a restricted endonuclease, which is a type of enzyme that specifically cleaves it at certain locations. It doesn't just randomly, it's very specific as to where it cleaves it. All right, and it's kind of showing you that right there. So here is our DNA from bacteria, and you treat it with a very specific enzyme that cleaves it at certain places. All right, so what it does is it opens it up, okay, into a long strand, into a linear strand, okay, and the two ends are said to be called sticky. It's because they have very specific, because of the enzyme used, it cleaves it at very specific sites. Okay, so for example, it's kind of showing you that you have an AA and a TT and a TTAA, so it's very specific as to what cleaved it. Okay. So then what you do is once you have that cleaved, all right, then a second sample of human DNA is cut from the same enzyme. So this enzyme now is treated with human DNA, and it's going to cleave it at the exact same sites that these were cleaved, okay, because this is very specific for certain sites on our DNA, or our bases, or our nucleotides, I guess, okay? So this forms a human DNA segment that also has sticky ends that match the sticky end over here. So when you take this, all right, our bacterial that you cleaved, as well as the human gene that has the same codes, and you treat them all together, these sites are going to perfectly find here, and they're going to match up and remake our DNA, hence the name recombinant. Okay, it recombines it. Okay, and again, it's a complementary. So a TTAA is going to find where you have an AATT. So this like segment right here is going to fit in right here. Okay. All right, so I guess I just kind of talked about it, but I guess what the third step is you take these pieces, you recombine them, you put a new enzyme, a DNA ligase enzyme, which it hooks it all together, and the DNA change is slightly larger just because you have like different size of our segments here. Okay, now that is how to make one of them. What if you want to make lots of these copies over and over and over again? Well, there's a technique called PCR, or a polymerase chain reaction, which amplifies a specific portion of a DNA molecule, producing millions of the exact copies. All right, basically, there are four elements that are needed to amplify the DNA by PCR. One is the segment of DNA that must be copied. Okay. Two primers, right? Short polynucleotides that are complementary to two ends of the segment that's amplified. So if you have a DNA strand, you just need short little primers that are the complementary match for the start end and the ending of it. Okay, and then a DNA polymerase enzyme to catalyze the whole reaction, and then finally you need all of the nucleosides, the A, the T, the C, and the G. Okay, so how does this work? You take the original DNA strand and you heat it up until the strand uncoils itself. All right, now you have two individual strands of our DNA. Okay, then you add the primers. What are the primers? All right, well, these, let's say 5 nucleotides are the complementary part of the 5 at our beginning, and these 5 are the complementary part at the end. Actually, here's the beginning, here's the end. So all we have is a beginning and an end. We don't need, obviously, a copy of the whole entire thing. All right. And what I'm sorry, the beginning of one and the beginning of the other. So you basically just have a few of them on each side. All right. Then you add in your nucleotides, so your A, your T, your G, and C, and you just throw them in there, and your DNA polymerase. And basically what the enzyme does now is it finds all of the nucleotides you have and it pairs them up like perfectly as our enzyme is going through. All right, and you basically will get an exact match. And it happens over and over and over again. Okay, now if you think about it, one strand just made, or like one DNA made 2 DNAs. Then these are going to unhook, and the 2 are now going to be 4, and those are going to make up to 16, and so forth and so on. And so you can imagine it's going to increase very, very quickly. So it shows you after each cycle, the amount of DNA is doubled. So after 20 cycles, you'll have 1 million copies of your DNA. Okay. Focus on the human body, how recombinant DNA is used in the human body. All right, the DNA of each individual person is unique, so DNA can be used as a method of identification. All right, any type of cell, skin, saliva, can be used to obtain a DNA fingerprint. The DNA is first amplified by PCR, and then it's cut by Bruch-Dictive enzyme so that you can figure out what the code is. All right, and the DNA fragments are then separated by size by what's called gel electrophoresis. All right, and what that basically is a gel. It's a medium. All right, and the DNA. So, and what you do is you kind of you put a charge, a current through it, and it kind of travels through. And all depending on the size of the fragment will either be, it'll either go a lot faster if it's smaller, or it goes a lot slower if it's larger. And so here's kind of an example of that. So of the fragments, these faster ones are the smaller fragments, and the larger ones, or I'm sorry, the slower ones are the larger fragments. All right, so let me stop there, and when we come back, we will talk about viruses.